Max Planck Institute of Colloids and Interfaces. The Max Planck Institute of Colloids and Interfaces German, Max-Planck-Institut für Colloid- und Grenzflächenforschung is located in Potsdam-Golm Science Park in Golm, Potsdam, Germany. It was founded in 1990 as a successor of the Institute for Physical Chemistry and for Organic Chemistry, both in Berlin-Adlershof, and for Polymer Chemistry in Teltow. In 1999, it transferred to newly constructed extension facilities in Golm. It is one of 80 institutes in the Max Planck Society Max-Planck-Gesellschaft. Research. Being part of the Max Planck Society, the institute examines nano and microstructures, specifically colloids, in which many are found in nature. With discoveries, scientists create tiny apatite crystals in bones, vesicles formed out of membranes, pores in membranes for fuel cells, and microcapsules as vehicles for medical drugs. All are larger than an atom, yet too small to be seen with the naked eye. The scientists at the Potsdam-based institute endeavor to understand how they are composed and how they work in order to imitate behavior in new materials or in vaccines, for example. Understanding the function of these structures can also help to identify the causes of certain diseases that occur when the folding of membranes or the transport of materials in cells fails to work properly. Colloid chemistry. The Colloid chemistry department, headed by Marcus Antonietti, deals with the synthesis of various colloidal structures in the nanometer range. This includes inorganic and metallic nanoparticles, polymers and peptide structural units, their micelles and organized phases, as well as emulsions and foams. Colloid chemistry is able to create materials with a structural hierarchy through appropriate functionalized colloid. This creates new characteristics through the teamwork of the functional groups. With appropriate architecture, these colloids can fulfill very specialized tasks. Single molecular systems cannot do this due to their lack of complexity. An example for this is skin. There is no synthetic material which is as soft and simultaneously saute-resistant, and yet is made mainly of water. The secret of this also lies in the interaction between three components: collagen, hyaluronic acid, proteoglycan. This unusual combination of characteristics is only made possible by forming a superstructure in a team. Biomaterials. The Department of Biomaterials, headed by Peter Fratzel, focuses on interdisciplinary research in the field of biological and biomimetic materials. The emphasis is on understanding how the mechanical or other physical properties are governed by structure and composition and how they adapt to environmental conditions. Furthermore, research on natural materials such as bone or wood has potential applications in many fields. First, design concepts for new materials may be improved by learning from nature. Second, the understanding of basic mechanisms by which the structure of bone or connective tissue is optimized opens the way for studying diseases and, thus, for contributing to diagnosis and development of treatment strategies. A third option is to use structures grown by nature and transform them by physical or chemical treatment into technically relevant materials by biotempering. Given the complexity of natural materials, new approaches for structural characterization are needed. Some of these are further developed in the department, in particular for studying hierarchical structures. Theory and Biosystems. The Department of Theory and Biosystems, headed by Reinhard Lepowski, investigates the structure and dynamics of molecules, colloids, and nanoparticles in biological and biomimetic systems. The molecular building blocks of these systems assemble by themselves and form a variety of supramolecular nanostructures, which then interact to produce even larger structures and networks. These complex processes represent hidden dimensions of self-organization since they are difficult to observe on the relevant length and time scales. Current research focuses on molecular recognition, energy conversion and transport by molecular motors, dynamics of transcription and translation, as well as self-organization of filaments and membranes. Interfaces. The Department of Interfaces, headed by Helmuth Meroldt, is primarily motivated to understand molecular interfaces and to relate this to colloidal systems which are by nature determined by the large surface/volume ratio. Consequently, the strength of the department in characterizing planar or quasi-planar interfaces has been increased, and in addition it has been tried successfully to transfer this knowledge to curved interfaces. From this we have again learned about planar interfaces, since surfaces could be studied by techniques requiring large surface area NMR, DSC. Biomolecular systems. The researchers in the Biomolecular Systems Department, headed by Peter H. Sieberger, are using new methods for synthesizing sugar chains. Until recently, most of the known naturally occurring sugars were those that supply energy to organisms such as sucrose, household sugar, and starch in plants. However, the complex sugar molecules, which belong to the carbohydrate, are also involved in many biological processes. They cover all cells in the human body and play a crucial part in molecular identification of cell surfaces, for example, in infections, immune reactions, and cancer metastasis. Complex sugars are omnipresent as cell coatings in nature and can therefore also be used for vaccine development, e.g., against malaria. Carbohydrates are thus of significant interest for medicine. The major significance of sugar residues on the surfaces of cells for biology and medicine has only been recognized during the past approximately 20 years. Until recently, a chemical synthesis method to create biologically relevant carbohydrates with a known structure in large quantities and for biological, pharmaceutical, and medical research was lacking. Now, these gaps can be closed with the development of the first automated synthesis apparatus that can link sugar molecules with other sugars or also molecules. Organization. The Golm-based institute has a total of 358 employees, including 91 scientists and 99 junior scientists and researchers, 6 apprentices, 138 employees paid with third-party funds, and 24 guest researchers. The Institute of Colloids and Interfaces is currently headed by the following people. Scientific members, Directors Prof. Dr. Marcus Antonietti, Professor Dr. Peter Fratzel, Professor Dr. Ryan Hodlepowski, Professor Dr. Peter H. Sieberger, Director Emeritus Prof. Dr. Helmut Merwold, Administration Manager Andreas Stockhurst, Board of Trustees Ulrich Buller, Senior Vice President Research Planning, Fraunhofer Gesellschaft Ralf Emmermann, Vice Chairman of the Board of Trustees, Geo Verschanzed from Potsdam GFC Detlef Gunn, Chairman of the Board of Trustees, Chairman of the Board of the Charité Universitätsmedizin Berlin Norbert Klant, Member of the European Parliament Jan Jacobs, Mayor of the City of Potsdam Wilhelm Kroll, Secretary General of the Volkswagen-Stiftung Sabine Kunz, Minister of Science, Research and Culture, Brandenburg Wolfgang Plisch, Member of the Bayer AG Board, Robert Seckler, University of Potsdam. International Max Planck School on Multiscale Biosystems. The International Max Planck School Olympus on Multiscale Biosystems is a graduate program in association with the University of Potsdam, Free University, Humboldt University in Berlin, and the Fraunhofer Institute for Biomedical Engineering in Striedenberg. In this program, young talented doctoral students can work on a challenging research project and develop their scientific communication and management skills. Scientific scope. The IMPS on multiscale biosystems addresses the fundamental levels of biosystems as provided by macromolecules and aqueous solutions, molecular recognition between these building blocks, free energy transduction by molecular machines, as well as structure formation and transport in cells and tissues. The research activities are focused on 4 core areas: molecular recognition of carbohydrates, interaction of biomolecules with light-directed intracellular processes, directed shape changes of tissues.